Membrane receptors are crucial for most processes in the body to be carried out to completion. Defined, a receptor is a structure that has the ability to respond to stimuli, such as heat or light sources, by sending a signal to a different part of the body. There are also different receptors in different cell types. How each cell responds to stimuli is called cellular response. The concept of membrane receptors can easily be demonstrated by looking at the phenomenon of signal transduction and how it relates to G proteins and cyclic AMP. This process is often identified by the indirect effect it has on the membrane potential. These indirect effects are commonly referred to as metabotropic. The process begins with a ligand. This is the messenger that begins the transduction process. It lodges itself in the receptor. This receptor is called the heterotrimeric G protein. This protein acts as a sort of bridge for the ligand to begin the reaction. Near the bottom of the G protein are the alpha, beta, and gamma subunits. When there is no ligand sending a signal, there is a GDP molecule attached to the alpha subunit. The unit will bind when the receptor has a ligand. When they associate, the GDP will be released and replaced with the GTP. The unit will then be able to send messages to other proteins. When GTP is changed back to GDP, the process must start over. In signal transduction, when the ligand begins the process, the alpha subunit transfers to the adenylyl cyclase further along the bilayer. When the alpha subunit connects with the adenylyl cyclase becomes an enzyme that has the ability to change ATP into cyclic AMP. These cyclic AMPs are now able to disperse throughout the cell. The CAMPs, for short, are looking for something called protein kinase. The CAMPs attach themselves to the protein kinases and separate into two parts, a catalytic part and a regulatory part. From this, the catalytic parts are phosphorylated by leaching energy from the ATP molecules and are then able to do work on other enzymes. In some cases, these activated catalytic parts can also trigger the enzyme phosphodiesterase. This enzyme will break apart any phosphodiester bonds, which will in turn create a reaction within the cell. This process often alters the protein conformation of some parts of the cell, as the sequence is being disrupted. Protein conformation is simply the structure of a protein and how it is put together.